All right, let's make urine, people. That's what's going to happen in lecturette 2. We're going to make urine, and the recipe for urine is to filter, reabsorb, we can do a little secreting, and then we concentrate. Let's do a quick overview of how urine is made in the body. The first step is the filtration of blood, which occurs in the renal corpuscle. And as blood flows into the glomerulus, water and small molecules leave the bloodstream and enter the Bowman's capsule. The second step is reabsorption. Now, reabsorption is necessary because when we filter, we don't discriminate. We get rid of everything that is small, which are both waste products and nutrients we need, like water and sugars and so on. So in the reabsorption phase, we get all the useful products out of the nephron and bring it back into the bloodstream. The third step is tubular secretion. And in this process, it allows the body to directly secrete waste products into the nephron. And then lastly, we concentrate urine. And in this step, this is the last chance we have to reclaim water. Now let's go over each step of urine formation in more detail. So the first step of urine formation is glomerular filtration. And the amount of filtration that occurs at the glomerulus is driven by blood pressure. So the higher the blood pressure in the glomerular capillaries will result in a higher filtration rate. And this concept was covered when we did the blood vessel lecture. Okay. Now let's look at the contents of the afferent arteriole. Generally, we would have small waste products such as urea, carbon dioxide, and environmental toxins in the bloodstream that we need to get rid of. But we would also have small nutrient molecules like sugars, water, amino acids that need to be transported to cells in our body. The thing is, when we filter, anything that is small will be pushed out of the spaces between the endothelial cells that line the capillaries, and they become part of the filtrate. And there's no way to differentially filter small waste products and small nutrient products. What determines what gets filtered is size. And if you're small enough to fit between the tiny spaces between the endothelial cells, out you go into the Bowman's capsule. So what happens then is that results in a higher concentration of larger proteins or colloids traveling in the efferent arteriole because they're too big to be filtered, which will then play a big role in colloidal osmotic pressure, which I will discuss in a little bit. Now, something to point out is that cells are also too large to be filtered. So if you have blood in your urine, you need to see a doctor. And there are many different reasons why you can have blood in your urine, and those reasons can be from bleeding from the ureter's bladder or the urethra. But bleeding from the kidneys is not good because it means that you have ruptured your glomerular capillaries. And one of the ways you can rupture your glomerular capillaries is if you have chronic high blood pressure. Now, if you remember from the blood vessel lecture, the arterioles play an important role of providing resistance, especially to capillaries, because if you have too much blood entering the capillaries, you can rip them apart. Capillaries are very, very fragile. Now, before we embark on our journey through the tubular reabsorption, I want to share with you my strategy. And the way I teach tubular reabsorption is foundational. And what that means is that I'm not going to go over every single mechanism of how every single molecule gets reabsorbed. I'm just going to go over the main players with the hope that in your future, when you do need to know more details, it will be easier for you because the foundation has been laid down solid. Okay, now here we go. Stage 2 of urine formation is tubular reabsorption. And we use the term reabsorption because we've already absorbed these molecules in our digestive system. Okay, so what's going to happen now is we're going to kick everything out in the glomerulus, yes, in the renal corpuscle, and then we have to reabsorb everything that we need back into our body. Okay, so reabsorption means we are absorbing items that we have already absorbed previously. Okay, so we're going to start at the renal corpuscle, and we're going to do a quick anatomy review. And here, the renal corpuscle is made out of two structures, and we're going to have this first structure here. This is going to be the Bowman's capsule, and it is a bowl, and it's connected to a structure here, which I'll label later. And then we have our blood vessels. So starting on this side, we're going to have our afferent arterial. Okay. This section here, this is our glomerulus, which is a fenestrated capillary. So remember, there's slits in here, yes? And then this is going to be our efferent arterial. Now, as blood comes in, yes, so I'm going to put an arrow. This is our blood flow. Comes in through the afferent arterial, yes? Comes in. It's going to enter my capillary bed, the glomerulus. So remember that these spaces are small enough or large enough to let small molecules and small proteins, which can be also enzymes, yes? So, some small proteins, okay, into the bowl, the Bowman's capsule. So these small molecules and small proteins, enzymes, are going to be pushed out via hydrostatic pressure, okay? And then when the small molecules now leave and enter the Bowman's capsule, they're going to be called filtrate, okay? Now, the filtrate is going to flow. It's going to flow through the entire nephron, right? A whole bunch of tubes that we're going to draw out one by one. And the first part of the tube is something called the proximal convoluted tubule, right? And we're going to abbreviate it as PCT. And the PCT is selectively permeable, which means that cells that line the PCT will allow certain molecules to enter the cell from the lumen, okay? And if you think about that word, selectively, what that means for the cells that line the PCT is they need to make specific channels and carriers in order to reabsorb certain molecules. Now, let's review reabsorption and what that looks like. And we've done this a few times in class, but let's just review none the same. So what I'm showing you here is this is going to be a nephron cross-section. Okay. And I'm just going to draw you the tube, but I'm drawing you the tube big, right? So this is a big tube here. Okay. Okay. And then the tube is lined with cells. So I'm going to draw my 3D structure here. Okay. And this is all cells. Now, in this area here then, if this is a cell, out here is going to be my extracellular matrix, otherwise known as the interstitium. Whatever word you want to use is fine. They all mean the same thing. And then, out here in the periphery, this is going to be my capillary bed, okay? Because all tissues have capillaries right next to them. Has to be. That's how the body is made. This is our conduit to nutrients. Okay, so let's label this capillaries. Okay, now this area here, this is the lumen. Remember, the lumen is an empty space, okay? Empty space of a tube. and it's gonna contain filtrate. What does that mean? Small molecules. Okay, so I got some sodium in here. I got water. I got chloride ions. I have calcium ions. I have small enzymes. I have small amino acids. What else do I have? Oh my God, glucose. Can't forget glucose. Glucose. Okay? So remember, what happens at the renal corpuscle is hydrostatic pressure causes anything that can fit through those slits to come out. There's no discrimination. If you need it, if it's important, oh, I'm sorry, too bad. It's small. So it's coming out of that space. Okay, so what the nephron needs to do in different areas of the nephron is we have to reabsorb everything that is vital for life. Okay, so we already know sodium is vital for life. So let's look at that. So this is sodium right here. In order for me to get sodium back into the cell, reabsorption, I need to get sodium first into the cell that is lining the proximal convoluted tubule. Okay. That's one compartment. Remember this compartment business, okay? And then sodium has to go into the extracellular matrix, okay? What's the other compartment? The blood vessel. I get sodium into the capillary. It is now reabsorbed because once it hits the bloodstream, it can now circulate throughout the body. So at the proximal convoluted tubule, what we're doing here is now reabsorbing about 70% or 80%. It really depends on what textbook you're reading. You don't need to memorize that number. Just as long as you understand is majority of the reabsorption of the molecules we need to sustain life is happening at the proximal convoluted tubule. All the glucose we kicked out, all the salt, all the amino acids, water, some calcium, everything we need is getting reabsorbed at the proximal convoluted tubule. Now, do something escape? Yes, because we can't reabsorb that fast. And we'll take care of reabsorption as we go through the rest of the tubule. Okay. Now, before we go on to the next section of the tubule, I want to show you folks one of the mechanisms to allow us to have reabsorption. And it is one that we learned at the beginning of the semester. Okay. Look at this. Here is a slide that is depicting now if this is glucose, yes, look at how glucose is coming into the cells that's lining the PCT. This is a symport. Okay, and so I have glucose come in and sodium, and then I can now have glucose traveling down its concentration gradient into the extracellular matrix, and then I just maintain the sodium gradient via a sodium ATPase. But look at the arrangement. It allows me now to reabsorb glucose and sodium at the same time. Okay, the process is the same for amino acids. I have sodium glucose, instead of a sodium glucose symport, I have a sodium amino acid symport. Okay. Now let's keep reabsorbing, and the next structure we're going to get into is the loop of Henle. Okay, so I'm going to draw it here for you folks, and I'm going to draw a big loop. And if you don't remember this from anatomy, this is the loop of Henle, and it has different parts of it, but I'm only going to talk about the descending loop. That's what DL stands for, and the ascending loop. That's what AL stands for. Okay, so the descending loop of Henle is semi-permeable. And what that means, if you remember from midterm 1, is water can cross. Okay, so what's happening in this area is this is where I have the reabsorption of water. Okay, now if I'm going to reabsorb water consistently, I have to have a protein channel. Yes, because water is polar. And I did tell you folks at the beginning of the semester, water can cross in between cells, or in between cells, but also in between the phospholipids within a cell, but only in small amounts. So here in this area, I have something called resident aquaporins. Okay. And what that means is I have a water channel that's permanent and it stays there. And this is what the water channel looks like. Okay, so here it's called an aquaporin, which means hole for water. It's a channel, okay, and it allows water to cross down its concentration gradient. Okay, so let's draw what's going on in here. So here I have water now coming out. This is water. I have water coming out and getting reabsorbed. Now, as water leaves, okay, what's going to happen now in the loop of Henle as I start to descend, yes, is that sodium is going to start to become more and more concentrated. So let's start to draw sodium. So here I do have some leftover sodium after I leave the proximal convoluted tubule. Remember, I can't reclaim everything all at once. I gotta, I need some more nephron to make that happen. So as I now go through the descending loop of Henle, what's happening with sodium is, because I'm losing water, the concentration of sodium is getting bigger and bigger and bigger. Okay, and that's what I'm trying to show you here with this big writing in this orange pen of my daughters. Okay, so sodium concentration is increasing because I'm losing water, right? Much like if you reduce a liquid, if you're making soup and you want it to be flavorful, boil all that water off, okay, because all the solutes remain behind. Now, what's going to happen in the ascending loop? The ascending loop is selectively permeable. Okay, here's that word again. And it's selectively permeable for sodium. And that's where sodium reabsorption occurs. So now what's going to happen is as I go up the loop of Henle, which is the ascending loop of Henle, there's going to be resident sodium channels in here. Okay, that's going to allow me now to have sodium exiting. Sodium exiting. Okay, to the point where now when I get to the top of the ascending loop, sodium concentrations have now gone back to what they were originally before the loop. Okay, so I want to show you folks something. Now, water moves down its concentration gradient. You folks also know from our osmosis lab that solutes pull water, aka solutes suck. Okay, so here, if you look at the loop of Henle, this is a beautiful design, because what is happening here is one side of the loop, water is coming out because it's attracted to sodium that's coming out from the other side of the loop. So both sides of the loop are creating conditions where the other can come out. Water is attracted to the sodium, and sodium is now traveling down its concentration gradient. Okay, now let's move on to the distal convoluted tubule. Okay, and so this is abbreviated as DCT. And here, let's draw this wiggly line for us because convoluted is curvy. Now, the distal convoluted tubule is selectively permeable or impermeable. Okay, so in this area, we can either reabsorb sodium or not, depending on what's going on with our body. Okay, so let me run some physiological examples to you. Okay, now you folks know I like salt, and if you had me for anatomy and now you're having me for physiology, you know I love nacho Doritos. Okay, now if I just went to Costco and I pounded the hugest bag of nacho Doritos. And if you folks don't know what nacho Doritos is, I'm sorry to be exclusive. Let me include you into my love. Nacho Doritos is a very, very salty fried chip is all. And it has this orange salt dye. Not good for you, but it tastes amazing. But if you ate a Costco-sized bag of nacho Doritos, you're going to have a lot of salt in your body. So if you have a lot of salt in your body, do you want to reabsorb sodium? No, you want to pee it out, okay? So if I have high salt in my body, what's going to happen in the cells that line the DCT, they will become impermeable to salt. Now think about what that means, people. What has to happen mechanistically in order for you to become impermeable to salt? You must first ask yourself, what is salt? Okay? What is salt? Go back. This is all exam 1. Salt is an ion, a positively charged small ion. Therefore, you know the movement of salt, sodium, you need to have a channel. So let's say that I don't want to reabsorb sodium because there's too much of sodium inside of my bloodstream. Then what's going to happen is all the cells lining the DCT are going to remove the sodium channels from their cells. Yes? Okay. And then what's going to happen is that those sodium molecules are going to continue on down the piping, and then we're going to urinate them out. Now, let's say that I've restricted myself from all salt because now the doctor says I have high blood pressure because I've been imbibing too much Doritos. Okay. Now what's going to happen is I need salt. I need salt, people. I need sodium to run every membrane potential in my body. Okay. You can die without salt. Okay. So salt is a necessary vitamin. Okay. Or nutrient. And so what's going to happen now, if I need more salt, is that the cells in this area are going to go ahead and put up more sodium channels. And then in this area, then if I have more sodium channels, then I can reabsorb. I can reabsorb sodium because I need it. Okay. And then that sodium can now get reabsorbed into the bloodstream. So that's what it means to be selectively permeable or selectively impermeable to sodium. Okay. I control whether there is a sodium channel on the cell membrane, and I control it via hormone secretion. Okay. So for now, I'm just going to say that the DCT permeability is controlled by the hormone called aldosterone. All right, let's talk about the last part of the tubing in our kidney, and this is something called the collecting duct. Okay, so let's draw it first. So the collecting duct is the last portion here that I'm drawing, and all distal convoluted tubules will feed into a collecting duct. Okay, and so a collecting duct can either be semi-permeable or impermeable to water. Here's that wording again, okay? Water reabsorption if needed. So let's run 2 physiological scenarios to you, okay? So back in the day, there was this competition online that you could wee for a wee. And what that meant was WEE for a WII, okay? So you could wee or pee, and if you peed a lot and drank a bunch of water and pee a whole bunch, you could win. Not Nintendo. Is it Nintendo Wii? Yeah, Nintendo Wii. It's a game. Okay, it used to be expensive. It used to be the thing to have. So a bunch of contestants pounded a lot of water, and they actually died So don't do that. But the point I'm trying to make is what's going to happen if you drink a lot of water, okay? So if you drink a lot of water, you're going to have a lot of water in your bloodstream. You're going to be filtering out a lot of water in the glomerulus, and in the nephron, you're going to have a lot of water, okay? So the job, or one of the jobs of the collecting duct, is to reabsorb water if it's needed. So if you have a bunch of water in your system, do you want to reabsorb all that water? No. No, you don't. And so what's going to happen is the cells of the collecting duct will not put up aquaporins, okay? And so therefore, water will come right out. And then later, collecting duct will go into different portions of the kidney. And once it leaves the collecting duct, it is now technically urine. It's no longer called filtrate, okay? Now, let's say that you're walking through the desert. Oh, no, you're walking through Mountain House on our campus during the summer, and it's hot and there's no water. Okay, it's like 100-something degrees. It's hot, hot, hot. Okay, you don't have anything to drink, and what do you think you're gonna do to the water that goes through the nephron? Are you gonna pee it out, or are you gonna reabsorb it? Reabsorb it. Okay, so what's gonna happen at the collecting duct is I'm gonna now put up. Yes, all the cells in this area are gonna put up aquaporins to let water come back into the body, okay? Because the collecting duct, this is the last chance for romance, people. If you don't reabsorb stuff in the collecting duct, it's done. Once it leaves, it's urine. We can't reclaim urine. Humans can't, because we're inferior animals. Other animals, desert animals, can. But for us, once you leave the collecting duct, you're done, okay? So we can now have this selective control via another hormone. And this hormone is called ADH. Okay? So collecting duct permeability is controlled by a hormone. And this hormone is called anti-diuretic hormone. Okay? And now words are important, so let's dissect this word. What does anti mean? It means no or against, okay? What's a diuretic? Something that makes you pee. Alcohol, coffee, okay? So this means the no pee hormone, okay? So think about it. If you are now secreting a bunch of ADH, you should be asking yourself, what's the condition in the collecting duct? And then also ask yourself, what's the condition of the collecting duct if I'm not secreting the hormone? You folks think about it on your own, and then that will help you folks brain grow. Okay, now that we've reabsorbed, let's talk about secretion. And so before we go into secretion, we have to define and review some terminology. So as a reminder, okay, when we talk about excretion, okay, what this means is I'm going to remove material from a living organism. So think about what that means. That means that whatever we're excreting is going to leave living tissue. It's going to leave the body, okay? But when we talk about secrete, okay, so secrete is a little bit different. Secretion, all that means is I'm going to move material or molecules, whatever you want to write in there is fine. I move material from point A to point B. Okay, so I just wanted to clarify that before we move forward. Now, in regards to secretion, the kidney is an amazing thing because now what we just talked about originally is, hey, let's make a filtrate that's now having filtrate or small molecules leave our circulatory system and go into the lumen of this nephron. But there's another entryway. Our body, for some molecules, not all, okay, in order to get rid of toxins, we can directly secrete it from the plasma, and this is from blood vessels that are residing here, okay? They're residing here. So from the plasma straight to the lumen of the nephron, okay? Now, this is the reverse of reabsorption, okay? So here, what we want to talk about is how do we get these molecules to go from our bloodstream? We're bypassing the glomerulus. We're going from our bloodstream straight into the lumen. And there's special carriers for foreign substances, okay? So I don't want you folks to memorize the names of these carriers. They're called OATs and OX, okay? So they're transporters that can take organic material that's negatively charged and positively charged and then just put them straight into the lumen of the nephron. Now, this plays an important role in regards to medications. So for some people, and you may know some people are very sensitive to medication, but some people are not. And so let's talk about opioids, right? This has been a running topic for this semester. So for some individuals, they can take opioids, but they don't really feel their effects. And there can be a number of reasons of why. The most popular reason is because an individual has built up tolerance to opioids, meaning that your body is clearing through the liver. And we'll talk about this in the digestive system, but the liver is your detox system. Okay, so what the liver can do now is take all of these molecules, right, that are toxics or toxin to our body. The liver makes enzymes to them, degrades them, and kicks them out of the body. Okay, but also, there can be these transporters that are residing in the cells that line the nephron, and they will allow us now to have, let's say, opioids just boop, go into the nephron and we pee it out. So some individuals are built with a lot of these organic transporters to where they become very insensitive to the medication, and it has nothing to do with tolerance. It's because they have these transporters in high amounts. Okay, now moving on to concentrating the urine. And so we've already talked about this. I did a quick overview, and we're just going into a little bit of a review and a little bit more details. And so here, when we concentrate the urine, this is what's going on. So there's this terminology here. I'm going to go over it in this picture. So this is the loop of Henle, okay? So this is part of the proximal convoluted tubule. This will become the part of the distal convoluted tubule. And there's something called the counter current multiplier of the nephron, okay? And all that means is the following. And you already had this, okay? So this is review. So here, I do reabsorb some salt in the proximal convoluted tubule. You folks know that already, all right? Now, if I start off with 300 milliosmoles of salt in the proximal convoluted tubule, you know that that concentration is going to start to increase of salt because what's coming out of here? Yes, okay? Water. Water's leaving here, and this is passive. Okay, there's no energy required. It's passive, and because water is now attracted to the solutes in the interstitium. Okay, now I've lost all this water. Look at this concentration of salt, 900 milliosmoles. But when I come around, right, what's going to happen is because there's more salt in the lumen of the nephron than in the interstitium, salt is going to leave down its concentration gradient. Bam! All into the periphery. And you see now, I had showed you in our drawing that salt and water are attracted to one another. They create gradients so one another can leave without costing any energy. There's no ATP needed for this scenario. And in doing so, as sodium comes out, I can have another place where I reabsorb water and reabsorb sodium, both necessary for life. And that allows me now to have the concentration of sodium start to decrease again as I get into distal convoluted tubule. Now, I already showed you, if I need to reabsorb more salt, let's say I'm extremely salt deficient, you already know what's going to happen in the distal convoluted tubule, okay? I can have a hormone called aldosterone, which we're going to call Aldo from here on out, A-L-D-O. Aldosterone can cause the permeability of the DCT to change. And if you remember, if I'm going to become impermeable to sodium. I cannot have any sodium channels there. But if I want to be permeable to sodium, then I'm going to go put a whole bunch of sodium channels on the cells that reside here, and that will allow me to get sodium back into the interstitium that will then go into a capillary where I can now get it back into the body. This is a beautiful, beautiful design. It's not regulated, people. It just goes and goes and goes on its own, all based on concentration gradients. That is a beautiful thing. Now, we got to maintain that concentration gradient, don't we? This has been a reoccurring theme in our class. This water and salt can't just sit here, because what's going to happen to the concentration gradient? We're going to reach equilibrium. So if you folks remember from the anatomy review that we did, is around the loop of Henry is a special capillary bed called the vasa recta. Okay, so the vasa recta, and all that means is the vessel that stands straight. Okay, and so here's the vasa recta, and its job is to maintain the extracellular fluid in regards to concentration gradients for salt and water. Okay, so what I want to do for your folks is, for this slide is the following. Okay, now I don't want you folks to memorize what part of the vasa recta is taking up salts and water. Okay, all I want you folks to understand is that the vasa recta is responsible for the following. Okay, so the vasa recta, and remember, what is the vasa recta? Capillary bed around the loop of Henle. Okay, so what it can do now is maintains the concentration gradient of the extracellular fluid or matrix or interstitium surrounding the loop of Henle so water, that's a descending loop, and sodium, that's the ascending loop, may continue to be reabsorbed. Whoops. Reabsorbed. This is just another beautiful picture of the nephron and showing you all the concentrations of the filtrate as we move through the nephron. Don't memorize these numbers. But you can see now as we're getting to the collecting duct, you folks remember, it is also an area where I can have variable permeability. And anytime I have a variable permeability, meaning that I can shut off or turn on permeability for reabsorption or not having any reabsorption, then I can now control what's going on here via hormones. Okay, so what's going on in the collecting duct is the following. Okay, now here's this ADH, antidiuretic hormone. Okay, so it is made in the hypothalamus. It gets stored in the posterior pituitary. Okay, this is a posterior pituitary, right? And what's going to happen is when antidiuretic hormone is released, it's doing the following. Here's the mechanism of action. ADH will bind to its receptor. Oh, look, it has a cyclic AMP second messenger system. But what it does eventually is to allow you to get calcium into the cell so I can now exocytose aquaporins. These are all aquaporins, people, okay? So let's exocytose aquaporins into the cell membrane so I can make the collecting duct cells permeable to water. Yes? And so here, in doing so, if I got a bunch of water in the lumen and I'm passing through the collecting duct and I need that water, The more aquaporins I put on the cell membrane, I can have water come in, I can have water go back, and this is my extracellular matrix. So now I have water here, and then that water can go back into systemic circulation. All right, that's it for this lecturette. Coming up next, we're going to do how we regulate glomerular filtration rate.